trout Oncorhynchus micis. Doctor, the stage is yours now.
Thank you very much. Can you see my screen?
Yes, doctor. Yeah.
Just 1 second. There we go. All right. Thank you very much for the kind presentation. Thank you for giving me the opportunity to present. This is my first time in this meeting. Here we have a small work we're doing with fish and trying to use different byproducts from the Chilean different industries, in this case, wine industries. So we're looking at the dietary inclusion of encapsulated grape mark extract and its effects over growth and antioxidant markers in rainbow trout. We live in a demanded world, in particular in terms of natural resources. If we look at this figure here, we can see that several planetary boundaries are already in red, and in particular, from a nutritional point of view, the one that has to do with nutrient flows between different compartments is of our concern, right? Because some of these nutrients are already over the capacity of the planet to compensate and regulate their flows. The aquaculture industry is not an exception for that. And this industry is projected to grow more than any other animal production industry, in particular due to their use of natural resources that at some point is more efficient than farm terrestrial animals. However, this growth and the ability of the aquaculture industry to provide the food system with resilience is also questioned sometimes because the use of these natural resources, in particular fish meal, have been proven to be unsustainable. So this industry has been lowering the level of marine ingredients, not only fish meal, but also fish oil, to try to attain this sustainable fooder and be able to contribute to the resilience of the food system. This is also a tendency that can be seen in other animal industries. So we're going to start with that. In any animal industry, including aquaculture, we live in a world that is very complex in terms of different ingredients that we can use. Marine ingredients, animal byproducts, plants ingredients are the mainstream ones, if you want. But today also we have other kind of products, ingredients that are made of things that originally are not, like sawdust and other that can be used by single cell meal and other stuff, or the meals derived from different type of worm. The concept here is to be aware that we need to maximize these cycles in terms of efficiency, because what we do in nutrition is move these three elements across different compartments. We move nutrients, we move energy, and we move water. So the aquaculture industry is focusing here because these terrestrial ingredients are a possibility to add to the sustainability by reducing these other ingredients here. However, there is plenty of literature that moving from this to that, right? From the fish meal to the plant protein, there are many problems that could be arise, particularly associated with anti-nutritional factors that are present in different plants, and which limits the maximal level of inclusion of these plant products, protein or oils, particularly in carnivorous fish. However, on the other side, from the same or other plants, we can have different bioactive compounds that, once extracted from their matrix, can be used to stimulate different components of the fish metabolism or physiology, obtaining positive responses in terms of growth, immune response, and other aspects. In this regard, and having the idea of this cycling of nutrient, water, and energy through the diets, and trying to move from a linear economy to a circular economy using probably an adaptation of the concept in circular nutrition, right? We're trying to use products that are originally considered as waste in order to re-extract or obtain product that can be useful for fish, right? Well, in this case, we're going to focus on grape marc, which is a byproduct of the wine industry. Chile has a very big volume of production of wine, generating a lot of, grape marc, which has several interesting characteristics. In general, it's composed of these parts: stem, skin, seed, in these proportions, and has approximate composition described there, which varies in particular in terms of protein or fiber. But the interest or the part that we are most interested in, because in this way probably ruminants can use it efficiently, but not monogastrics, there are the polyphenols, this secondary product of the plant metabolism that has been described with different biological activities related in many different models, mammals and humans, with several benefits to health and other aspects, in particularly also associated with anti-inflammatory and antioxidant properties, which are our interest today. So since we don't have much information about the use of this type of byproduct, we set ourselves with this hypothesis in which the generation of this grape marc extract microencapsulated, is a mistake there, we can use it in the fish of diets, in the diet of fish, sorry, in order to improve growth and plasma antioxidant capacity. So our objective was to evaluate the use of this byproduct, right, in this format, because, as I said, in fish, it's very difficult to use due to the level of fiber, in particular, in a direct way. And we evaluated changes in terms of productive performance and changes in antioxidant capacity in fish plasma. Well, to do this, we use this grape marc from the variety Cabernet Sauvignon, very abundant in Chile. We dry it out. We obtain it directly from a winery. Then we preparated the extract using a procedure which is incubating an amount of this grape marc in a proportion of 5:1 in terms of grape marc and water, with temperature and constant agitation. And we obtain this liquid that we afterwards dried via spray drying, with different proportions of this wall material, maltodextrin. We study this in order to see which one of these concentrations could be more efficient to be used and to transform this raw material in something that can be easily included in the fish diets. With that, we prepare these pellets via extrusion using this equipment. And usually in the aqua feed, these pellets are full of little spaces in their structure, which is the space that afterwards is filled out with oil using this vacuum coater. And in this oil, we included different proportions in these diets of the microencapsulated grape marc. That way, we don't alter the composition of the meals, or we don't expose the microencapsulated product to the temperatures that are usually really pretty high during the extrusion process. So as a first step, we try to include it we did include it in the oiling process. Regarding the fish, we use rainbow trout with an initial weight of 90 grams in 12 tanks in a recirculation unit at the University of Chile, with pretty standard conditions, very similar to what the industry does, with photo periods of 14 hours of light, 10 of dark. Fish were fed ad libitum. We were pretty sure of that because if you can see in this picture, we have this little tank here on each of the fish tanks that can collect the feed that the fish do not eat. So every time we see an excess there, we are pretty sure that the fish is eating at satiety. We fed the fish 6 days a week during 90 days, and the diets were randomly distributed in triplicate in each tank. This is a small diagram of what we did and at which point we took samples and we took measurements of the fish in order to monitor their growth and the different variables we measured during the experimental period. In terms of the encapsulation of the grape mark, we measured the size of the particles, we measured the yield, the encapsulation efficiency, the total content of phenols, and the antioxidant capacity via ORAC. For the fish, we evaluated, of course, feed intake, the percentage of body weight, the feed conversion ratio, which is the amount of dry feed for a certain amount of weight gain, the specific growth rate, the size of the liver and the weight of the liver, sorry, and its relation with the weight of the body of the fish, and the antioxidant capacity in plasma. All this was analyzed using the tank as the experimental unit, using 3 tanks per diet, 5 fish for all the per tank for all the plasma work, and then using this model in order to evaluate the results. So what did we find? In terms of size, this is the range in which the produced particles we produced. This is in agreement with the characterization of different spray drying techniques described in the literature. So we are okay with that. And we observe an increase a significant increase when we move from the 24 to the 36% of maltodextrin inclusion in the particles measured. In terms of yield, we saw a different trend with significant difference as a decrease here, with increasing inclusion of the maltodextrin. These values also are considered in a proper range in this paper, in which they describe different yield measurements. They indicate that over 50% is considered relatively efficient. And for other works like those presented here with grape skins and grape and olive pomace, the results are very similar in terms of yield using a similar level of maltodextrin. This is very important because we believe, well, important in the sense that this is giving us information to decide at which level of maltodextrin we were going to work, which for the moment 12% looks better than the others. This decrease in the yield, we believe, is associated with the fact that increasing the maltodextrin content, one could expect an increase in the final powder obtained in the process. However, there is a relationship between the amount of maltodextrin we use and the shape of the particles. We also did an electron microscopy just to show them the shape of the spheres. And we saw a difference there, and with that also we found out that the amount of powder we could retrieve from the equipment that is not attached to the bottle in a form of paste is increasing with increasing level of maltodextrin, which is explaining probably why the yield is going down with higher level of maltodextrin. In terms of the efficiency at which the extract was encapsulated, there were no, it was high, first of all, there were no differences in terms of the efficiency between the different level of maltodextrin inclusion. This was basically for us the main deal breaker in terms of, or the decision factor in terms of going for the lower level of maltodextrin inclusion, because there is clear here a dilution effect in terms of total phenol content and the oral capacity of the extract when increasing the level of maltodextrin. So considering the previous values and this reported here, we use this microencapsulated extract to put it in all the diets that we fed the fish. These results are also in agreement with previous reports in which you can observe a decline in the content of different biologically active compounds when encapsulated with increasing level of wall material. In terms of fish variables, this is very important. The first one, feed intake as a percentage of body weight, not as a total, as a percentage of body weight. So, and we didn't see differences between the treatments. This is important for us because this allows us to say that the differences observed in other variables are not due to an intake issue, but to a differential use at the metabolic or physiological level by the fish. Otherwise, if we were to observe difference, statistical difference here in feed intake, that could be a very noisy data set to interpret, because it always is going to be associated with different level of consumption. Well, in terms of percentage of body weight, at the first sampling point, we didn't see any differences. At the second sampling point, already we can see some difference here with the diet without the inclusion of the grape mark. These are all the vegetable diets, high level of oil and vegetable protein. This is a positive control animal diet, rich in fish meal, right? And at the end of the study, we see that these two diets, which are the vegetable diet with the inclusion of the grape mark, have a better percentage of body weight compared with diet with no inclusion, only with the capsule, right, with the wall material. These results are also in agreement with some other work, previous work, using different products derived from grape. The thing is, the results are not very easily comparable, because in these other studies, they use, for example, extract from seeds or only skins. We are starting from a product less processed, considering it could be more accessible for everyone, and it could be a better option in order to generate this type of product. In terms of the weight of the liver and the hepatosomatic index, we also see differences, particularly in liver weight by the end of the study, in which the liver of fish fed the fish meal diet and the plant diet with the grape mark extract have, in this case, significantly higher values and numerically higher values compared with the diet with no grape mark inclusion. A similar trend was observed with the hepatosomatic index, the relationship between the liver weight and the body weight of the fish. These results also are in agreement with the literature, in particular with the level of carbohydrate observed in our diets. Finally, getting to the specific growth rate, no difference at the beginning, some trends at 60 days, and by the end of the study, all fish fed the vegetable diets with the inclusion of the grape mark extract showed significantly higher values compared with the negative control diet, if you will, reaching plant ingredients. In FCR, the conversion ratio, also very important for us. Here, the lower the value, the better. So less amount of food is required for 1 kilo of weight gain. At the beginning, no differences, as relatively expected, right? Because it takes time sometimes to see these differences. Some trends here at 60 days, and lower values observed at least for the higher level of inclusion of the grape mark extract compared with the vegetable diet. Similar results have been reported before, saying that some of the effect of this type of component, in particular polyphenols, has to do with the differences or the effect they have of glucose homeostasis in fish, changes in microbiota. That's a description we did in this paper, right? We measure changes in microbiota, and muscle development described in larvae, and antioxidant defenses. So these are some of the processes, even when we don't know exactly the metabolism and the mechanism by which polyphenol act on fish physiology. These are some of the routes described. In terms of antioxidant defenses, we observe significant differences in terms of ORAC values in the plasma of fish, even early in the study, right? Here, at 60 days, the trend is similar, with differences between the lower level, not the higher level with the plant protein, and finally, only probably disappear this effect because, well, we don't understand very well if other antioxidant mechanisms are in action here, because even when this is a very good measurement, it's only one. There are other compartments, so to speak, of the antioxidant response of the fishes, the enzymes, or other elements that could be influencing this final value. We have to consider that this corresponds to the non-enzymatic antioxidant response of the fish. But only ORAC measures one compartment of that. The results, again, they are in line with the previous literature, indicating that you can observe different differential levels of antioxidant capacity in plasma postprandially. But in this study, since we measure more long-term, there are no long-term measurement in other animals. So this probably is one of the first ones, at least in fish, describing this difference in a more longer period of time. So our conclusions are very easy. Thank you. Luckily for us, the inclusion of grape mark extract improved trout growth parameters, and that is in association with improved in ORAC values in trout plasma. This is part of the literature used in this research. Thank you very much for your attention, and I'm absolutely open for questions if there is any.
Thank you for that insightful talk, Doctor. Now the stage is open for questions. I assume that you have no questions from the speakers, Doctor. Once again, I thank you for being a part of this webinar and hope to associate with our future events in person. Thank you once
Thank you.
again and have a great day, doctor.
Thank you.